Very good morning to all. Seeking the blessings of His Holiness Sri Shivratri Deshikendra Mahaswamiji, and on behalf of the fraternity of JSS Academy of Higher Education and Research, Mysuru, I welcome you all to this webinar, Microbiome Listen to Your Gut, organized as part of Special Interest Group on Placental Microbiome and Factors Affecting It. Our gut harbors an entire universe of microorganisms with its own unique identity. Did you know that the gut microbiome protects us from diseases, or that the microbiome is shaped and affected by the diet you take? The microbiome is a collection of genes of these tiny microorganisms comprising of hundreds of different bacteria, fungi, and viruses. This microbiome, which itself is an organ, affects our digestion, aging, immune function, cognitive function, and even our mood and behavior. As the world is on the brink of health crisis with the new COVID-19 pandemic and other lifestyle-related epidemics, it's time to listen to our body and, more importantly, listen to your gut against the fight for health and survival. This webinar, therefore, is organized to give a peek into the gut microbiome and its importance in healthcare. On this bright morning, we have with us our esteemed speakers, Dr. Yogesh, Scientist G, NCCS, and Principal Investigator, National Center for Microbial Resource, Pune. I welcome you, sir. Mr. Kumar Shankaran, CEO of Premier Microbiome and Genomics-based company, Lucene Rich Bio, Bengaluru. A warm welcome to you, sir. Dr. Mukesh Kapoor, Principal Scientist, CSIR CFTRI, Mysuru. Welcome you, sir.
Thank you.
We have with us our Dean, FLS and Director Research, Dr. Balasubramaniam, sir. A warm welcome to you, sir. We also have with us our Head, Department of Water and Health, Faculty of Life Sciences, Dr. Ravisha K.A. Welcome you, sir. As we are on this virtual platform, let the light of knowledge enlighten our minds, and we pray for the blessings of the Almighty for the success of this program. Before we begin these sessions, I would request our Director Research, Dr. Balasubramaniam, sir, to speak a few words.
Good morning to everybody. On behalf of JSS University and Faculty of Life Sciences, I welcome all the panelists for this webinar, which is arranged. This technology and everything shows that distance is not a matter for sharing our knowledge and everything. So I thank Dr. Annie Catherine for arranging this seminar, and I thank the HOD for permitting and the university for giving the facilities for this seminar. Thank you very much.
Thank you so much, sir. We also have with us our head, Dr. Ravisha K. I request him to give his opening remarks.
Very good morning to one and all. Invoking the blessings of His Holiness Sri Deshikendra Mahaswamiji. I pray the Almighty for the safety and well-being of everyone in this very bad time of COVID-19 crisis. My appreciation to the Special Interest Group team for choosing the most appropriate topic for the webinar. Friends, as you all know, we are not alone in our being inside every one of us are trillions of microorganisms, be it bacteria, virus, fungi, or other forms of life that are collectively known as microbiome. Different organs have different but distinct microbial inhabitants, but the group that has attracted the most attention in biomedical research is the gut microbiome. To understand the role of gut microbes in health and disease, scientists around the globe are investigating what makes a good microbiome. Iran Segal argues that collecting microbiome data would allow a deep phenotyping approach that could transform drug discovery. Health promoting probiotic species studies are yielding biological insights that might promote drug development. Many diseases, disorders are now supposed to be influenced by processes in the gut microbiome. Those include cancer, autoimmune disorders such as multiple sclerosis and autism spectrum disorder. The gut microbiome also strongly interacts with certain drugs, including some mental health therapeutics, and influences their effects. With evidence mounting on the gut microbiome's health significance, the synthetic biologists are looking to engineer the microbiome both at the individual species level and as an ecosystem, primarily to thwart the development of disease. Of late, there is growing public interest as to how the gut microbiome can be influenced, often focused on personal dietary choices. Microbiologist Peter Tonbach reframes this as a question not of which food will benefit our health, but rather what medical insights might be gleaned from the interactions between our gut microbes and what we eat. Ladies and gentlemen, I'm sure the discussion and deliberations during this webinar will help to arrive at meaningful solutions and show new vistas for exploring this interaction for the good of mankind. I thank the special interest group, the JSS authorities, and the participants for for providing the opportunity to be a part of this webinar. Thank you.
Thank you, sir, for your kind words. I'm sure the audience is very eager to listen to our speakers. So without taking much time, I would like to introduce the first speaker of the day, Dr. Yogesh. So you can go ahead and share your slides, sir. Yogesh, sir, you can share. You can share your slides, sir. Dr. Yogesh Shauche, Scientist G, National Center for Cell Science, and Principal Investigator, National Center for Microbial Resources, Pune, India. He started his career as scientist at NCCS, Pune, India. After working on different aspects of microbial, especially bacterial diversity of various ecological niches, his thrust area of research is now to understand succession of microbial communities in human gut during development and their role in health and diseases. He is working since 25 years in the field of microbial ecology, microbial molecular taxonomy, and biodiversity. He has published more than 200 publications in journals of relevant area and reviewed publications in international journals like PEMS Ecology, International Journal of Systematic and Evolutionary Microbiology, and Microbial Ecology. He is also on the editorial board of prestigious journals like Current Science, PLOS ONE, etc. In 2009, he was given a responsibility of the establishment of microbial culture collection, which is now known as the National Center for Microbial Resources. It currently holds more than 150,000 bacteria and fungi in its collection and is also recognized by the World Federation of Culture Collection. MCC is also recognized as an international depository authority and is the designated repository for Ministry of Environment, Forests and Climate Change, Government of India, under the Biological Diversity Act 2002. In September 2018, Department of Biotechnology extended the mandate of this repository to include antimicrobial resistant microbes. So before I hand over the platform to Yogesh sir, I would like to let the audience know that you have a question and answer option at the bottom of your screen. You can type in your questions in the question and answer box, and I will put across your questions to the speakers at the end of the lecture. Now I invite Yogesh sir to present his talk on the topic, Human Gut Microbiome, Indian Perspective. Over to you, sir.
Yes, thank you very much for this opportunity, and it's indeed a pleasure to give this presentation. And I mean, because of this COVID, we are having all these online presentations, and it has been a totally different experience now to make these presentations. There are some advantages of it and some disadvantages. Advantages that you can really have a very large number of participants. Now here I can see there are more than 200 participants. So that's an advantage of having such online talks, and participants from many faraway distances can participate in such events. However, the disadvantage is that there is no direct one-to-one interaction, and you really cannot judge how the audience is taking your talk. Like at times the participant may be just online and he may keep his audio and video mute and doing something else. So you won't know. So that's the disadvantage. Nevertheless, it's indeed a nice experience to give this talk, and what I'm going to talk to you in next half an hour, 45 minutes is what is the microbiome, its global scenario and the Indian scenario. So as it is said that the microbiome is the biggest thing in biology after the Human Genome Project, and as was mentioned some time back that it has extraordinary human health implications, and as again we'll see during the talk that microbiome is personal, and thus personal microbiome offers personal health solutions. So that's the advantage of microbiome, that it offers you personal specific solutions and also affordable solutions. So that's why microbiome has assumed a great importance in last decade or so. Now, if you see globally, there are multiple microbiome projects. Almost each country now has its own microbiome project which envisages mapping and looking at the differences in microbiome. So there is a Canadian microbiome initiative, there is human microbiome project in United States, then there is France has its own microbiome project, MetaHit is European Union project, Human Metagenome Consortium Japanese project, China has its microbiome project, Australia has Eurogenital Microbiome Consortium, and Singapore has Human Gastric Microbiome Project. So worldwide there are many individual microbiome projects. Now if you look at the microbiome, so basically what is microbiome? If you want to understand what is microbiome, it's in simple word, it means that genome of all the different kind of microbes associated with human body. So if you're talking about genomes, it is microbiome, and if you're talking about organisms, it is microbiota. So that is the difference. And if you look at our own body, microbes in numbers, they outnumber human cells by several folds. Estimates vary. The original estimate was 10 times more. It was said that for every human cell there are 10 microbial cells. Now it has come down. People say that it is 2 to 5. So for every human cell there are 2 to 5 microbial cells. And if you look at the number of genes, then human body contributes something like 22,000 genes to our body, whereas total microbiome contributes several thousand, millions and trillions of genes to our body. So that's the importance of microbiome. If you look at the different organs, I have listed out some of the important organs here. Gut harbors the largest number of microbes. It carries the weight of microbiome present in the gut is to the tune of 1 kg. Then the other important organs are nose carries something like 10 gram of microbes, mouth carries something like 20 gram of microbes, and nose you have typically streptococci, in mouth you have streptococci and Neisseria. Vagina carries 20 grams of microbes, major genus is Lactobacillus. Skin carries around 200 grams of microbes, and typically it carries Staphylococci and Corynebacteria. And total weight of microbiome in our body is around 1.2 kilograms. Skin deserves a special mention because though it carries, in terms of weight, it carries less microbes, but it is much more diverse. It is as diverse as the gut, because as one can envisage, the different parts of the skin have different type of microbes, because it doesn't require a great deal of intelligence to understand that a skin on the forehead which is oily and exposed to outside would carry different microbes than the skin of pora, which is again exposed but it is dry, so it would carry different microbes. And a skin of armpits or groins would carry different microbial community because it is not exposed to outside and it is moist. So that way then skin harbors a much larger diversity of microbes than one can imagine. And it has very interesting consequences. I'll not go into the details of that, but people have shown that somehow the mosquito bites are also correlated to the skin microbiome. So where do we acquire microbiome from? It is more or less accepted, though there is some debate on that, but it is more or less accepted that the baby in the womb is sterile, and it gets its first exposure to microbiome at the time of the birth. And if the baby is born by vaginal delivery, then it harbors, it gets inoculum from the vagina, and it carries Lactobacilli prevotella and Schnitzia. In contrast, if the baby is born by cesarean section, then it carries Staphylococci, Corynebacteria, Propionibacteria. I mean the organisms that are introduced from the hospital staff and the hospital environment. And as the baby grows, then the microbiome changes. After the ingestion of solid food, the microbiome changes, which gets stabilized by approximately 3 to 4 years of age. However, what is important to understand that these first 2 years are very important. And again, after the birth, it also depends on baby whether the baby is breastfed or it is fed on some other food. That also defines the gut microbiome, because the breast milk is again a large reservoir of microbes. It carries something like 1,000 species of bacteria, and even the polysaccharides, oligosaccharides present in the breast milk, they serve as a growth promoting factor for the growth of healthy bacteria in the baby's gut. So they are not for baby's nutrition, but they are to selectively promote the growth of good bacteria in baby's gut. And if the baby is not given the breast milk, then, and if you give some other milk, then again the organisms that develop in the gut are a different kind of organisms. And what has been shown by some studies is that this crucial period of up to 2 to 4 years has very important influence and on the subsequent health of the baby, because the babies born with cesarean section are known to have allergies, and also they are more likely to develop diabetes and obesity and coronary heart disease. So this particular period from birth to 3 to 4 months is very, very important in terms of microbiome for the development of healthy individual. And then once the baby in the adulthood, the microbiome is more or less stable up to 60-65 years. Microbiome is stable, and then it starts changing again. As I said, the largest number of microbes exist in GI system. Stomach, of course, harbors the least number of organisms, around 100 per ml. Highest density is found in colon, where it is 10 to 12 CFU per ml. And I already mentioned that the colonization begins at birth, developments in the early stage of life. Microbiome is almost unique to each individual. So it is gut microbiome, skin microbiome, they are very individual specific. And again, I'm not going to talk much about that, but this individuality of microbiome has resulted in some forensic applications, though never used in the court as evidence, but they have indeed been used for individualization. I'll again make a passing remark to this once again. And as was mentioned earlier, that the microbiome is altered in many of the metabolic disorders. So they carry out many functions in our body. Some of the important functions are they help in the development of intestinal tissue. They are important in epithelial turnover, motility, and blood flow. They modulate our immune system. Most importantly, they are involved in energy utilization and storage. They produce micronutrients and short-chain fatty acids. They block colonization with pathogenic bacteria. And today we understand a great deal more about how the intestinal bacteria affect the colonization by pathogenic bacteria, and how in turn pathogenic bacteria try to evade these mechanisms. We understand a lot more about this than what we used to understand earlier. And again, this was mentioned earlier that orally taken drugs, they are metabolized by gut bacteria to their active ingredients. And in many cases, it has been shown, especially there were studies in blood cancer, where it was shown that a drug which is not otherwise effective can be made effective in certain individuals by modulating the gut microbiota. So they play a very important role in drug metabolism and efficacy of the drug. So, I mean, the fact that microbiome is individual specific, it also means that it's a region specific also. So this is a picture, a kind of a global scenario where it shows how microbiome differs globally. So if you look at Canada, USA, the predominant microbes are different. In South America, they are different. In South Asia, they are different. China, Russia. So each geographic region, it has a different significant microbiome, and this is because of geographical influence. This is geographical influence on the availability of food material, and the food in turn affects the diet, and then because of the diet is known to alter microbiome, and it is also because of the genetic factors. So all these factors influence microbiome and alter the microbiome. So earlier it was thought that the factors that govern health, it was host genetics and lifestyle that governs your health. But now we believe that there is a third factor, microbiome, that affects your health. And what is understood is that if we look at the type of microbes that are present, there are symbionts, there are commensals, and there are pathobionts. So as long as they are in harmony, you have homeostasis and there is a health. If this balance is disturbed, then you have an increased proportion of pathobionts, and then this is dysbiosis. And this balance can be disturbed by a variety of factors: immune system, metabolism, genetic factors, mutations, or microbiota modulators like antibiotic treatment, or infection with pathogens, or environmental factors, dietary habits. All these affect your microbiome, and then there is a increased proportion of pathobionts which is associated with dysbiosis. This is what we understand today. So this association has been shown in variety of disorders like type 2 diabetes, cardiovascular disease, inflammatory bowel disease, non-alcoholic fatty liver disease, obesity, autoimmune disorders, metabolic syndrome, many cancers, and autism. So all these diseases, this association of dysbiosis and disease has been shown. And if you look at the number of publications that is coming out in microbiome space, it has increased exponentially in last couple of years. Everywhere you see 2012 and 2002 and 2012 comparison, you will see that the microbiome publications have increased several fold in this year. So that emphasizes the importance of the field. And 2017, the number of publication was around 40,000, and now I'm sure that number has reached around 60,000. Same is true with the patent. There were around 1,000 patents in microbiome space by 2017, and now again I'm sure the number has increased by 1,000 more. All important journals, Science, Cell, Nature, they have covered microbiome on their cover in the last couple of years, again to emphasize how microbiome research is important, and it offers many solutions. So it offers personalized solutions in microbiome, and so it offers you the prevention of the disease in terms of risk assessment, follow the population and risk, and early detection. And here, I mean, there are examples of this, but diabetes is one example. Again, I'll show you some more data on this later. But in diabetes, it has been shown that from the healthy individual to pre-diabetic to diabetic, there is a microbiome change. And then if this can be captured, even before the onset of diabetes, the microbiome change can be captured, then the onset of diabetes could be delayed and it can help in early detection of diabetes. There is a application in diagnostics also, so it offers you better diagnosis. It obviously offers you therapeutics, the use of microbes to treat the diseases. Again, I'll give you an example of this later. And personalized nutrition. So since diet and microbiome are correlated with each other, the modulation of diet will change the microbiome, and it offers you solutions to give you healthy gut microbiome and maintain your health. And just to give you an example of this, probably it's not there in the next slide. No, it's not there in the next slide. I'll just give you the example of this, that some 2 years back there was a study done in Israel. Here they had taken some 800 individuals, and there they had a mobile app where the person was expected to log in each activity, in the sense that walk, time of sleep, duration of sleep, any physical activity, plus the diet. I mean, each and every dietary component, the quantity as well as identity, they were supposed to log in using a mobile-based app, and there were skin pads. I mean, the sensors were introduced below the skin, and the blood sugar was monitored every 5 minutes. This was done for the entire week, and even the microbiome of these individuals was studied. So at the end of this experiment, they compile the data and analyze, and and one more important factor is that unlike in India, in Western population, the diet is more or less homogeneous. So there is not variation in the diet. Mostly the same food is, but items are purchased from the supermarket and they are just eaten at home. So they normally take cold meal also. There is very little processing of food at home. So because of that, all the commonly consumed dietary item, the nutritional values were known. So fiber content, protein content, vitamin content, everything was known. So when using this, then all this data, when it was analyzed, they found that the same food does not have the same effect in all the individuals. For example, if you just take banana, the rise in blood sugar level after eating banana was different in different individuals, and this change was correlated with the microbiome. So based on this data, they made a model, and they found that the model functions better if you bring in microbiome component into that. So point here is that the way food is metabolized in different individuals is different, and microbiome plays a very important role in metabolism of the food, and that's what is meant by personalized solution. So if one is recommending a food for management of diabetes or for good health, then there cannot be a generalized dietary recommendation. The dietary recommendation should be in considering the person's lifestyle and the microbiome and basic metabolism of the person. So all that need to be taken into account while making a dietary recommendation for the person. Now, if you look at the microbiome market, globally it is estimated that by 2024, the microbiome business will be in US alone, it could be 365 million dollars. So that's the globally that's going to be the microbiome based business, and this is going to be in different aspects of microbiome. So it is going to be IBS, cancer, IBD, then immune system disorders, hypertension, multiple sclerosis, diseases like Parkinson's disease and Alzheimer's disease. So all these are potential diseases have a potential scope for microbiome therapies. Coming to India, we all know that we are a very diverse country. We have 10 biogeographic zones. If you look at the entire country, we have 10 biogeographic zones which affect the productivity and also in turn the diet. And there is a tremendous dietary variation. Even within a single geographic variation, there is a single biogeographic region, there is a tremendous dietary variation. And the ethnicity also is varied amongst all over the country. We have something like 4,693 population groups and 2,205 major communities, and we have second largest population. So combination of all these three together affects the microbiome, and the variation in the microbiome across different biogeographic regions is going to be tremendous. So it's when we are talking about a microbiome based solution for the country, it's not going to be a single solution for the entire country. There has to be different solutions considering the biogeography, dietary habits, and the genetic makeup of that particular region. So these solutions have to be based on that, and a single probiotic supplementation or single microbe as a solution is not going to suffice for the entire country. That we should remember. So what we have studied is in my group we have studied microbiome in health and disease. So in health we have studied mother-infant microbiota, age differences, genome sequencing of isolates, and try to understand what is healthy Indian microbiome. In diseases we have studied diabetes, celiac disease, kidney stone, and inflammatory bowel disease. So in next few minutes I'll just give you a flavor of that. What are our salient observations? What we found is that there is definitely age-related changes in the microbiome. This manuscript is now actually accepted in scientific reports 2 months back, and so the important factor over here is that this study was done in the joint families. The advantage of joint families is that whatever 3 or 4 generations one is studying, they live in the same house and eat the same food and exposed to same environment. So that makes these studies easier, because the other factor like variation in the diet or environment don't come into the picture, and one can really study the age-related changes in the microbial population. And what we found was that with the age, the proportion of proteobacteria and fusobacteria increases in the population, and as against that, the bacteroids population decreases with the age. This is what we had observed in the population which we had studied. Now, the same thing we observed in oral microbiome also. We found that with the age, Treponema, Fusobacterium increases, and Streptococcus and Granulocartella decreases. So both oral as well as gut microbiome, there are age-related differences. If we look at globally, we did global comparisons with all possible regions. We did comparisons with Western population, African population. Then we also did comparisons with other Asian populations, and we found that the gut microbiome of Indian population, as we studied, is always different from the rest of the population. So here it was not a total India representative study. Here the majority of the individuals were from West. Taken some individuals from North, which we had used as a control in some of our other studies, but the picture remains the same. And what we found is that we also tried to do correlation between dietary components, but we did not get very good. I mean, we looked at the fiber, carbohydrates, this and that, and we tried to do correlation, but we did not get a very good correlation because this was a homogeneous population. The population which was studied here was a homogeneous population, and they all had the common diet. So when they have a common identical diet, then the correlation between different diet and the microbiome composition is not very good. So that's the disadvantage of this particular study. And also, I mean, I'll come to that point later. So then, talking about the diseases. Okay. Yeah, and another important thing is about isolates. So we tried to do isolations from some of the samples which we collected. We got more than 100 isolates from a single individuals, and out of these 100, at least 32 isolates had the potential of being a novel taxa, out of which so far we have published 7 novel taxa. We have 5 more in the pipeline, but there are some other issues. There are some other issues nowadays about publishing novel taxa. I don't want to go into those issues in this forum. But what is important here is that I forgot to mention that during my previous slides that the major constituent for in the human Indian gut is the Prevotella and Megasphaera. So Prevotella and Megasphaera are the predominant organisms in human gut, and we also found that during our isolations we reported Megasphaera for the first time from the gut of a human being. Earlier it was reported from ruminants. So talking about Prevotella, what I wanted to emphasize here was, though we found Prevotella in the gut, and the only gut isolate that has been cultured so far is Prevotella copri. But the organisms present in the gut of Indian individuals are very different from this Prevotella copri. So they are most likely to represent novel species or new species of Prevotella, or maybe even a different genus. So that is a point which I wanted to emphasize, that the organisms, though they are Prevotella, physiologically they would be very different from the already grown organism Prevotella copri. So that, and even, I mean, this is another point which I wanted to emphasize here, that even though at the 6' RNA gene sequence level, the organisms, even if they are very close, at the protein level or functional level, they could be very different. So we had isolated this organism from gut again, which we called as Clostridium punens. And what we found is that at 6' rRNA gene sequence level, it shares 99% homology with Clostridium tunisiense. But at the genome level, they are very different. And if you look at, for example, the genes that are responsible for carbohydrate metabolism, tunisiense, these genes are very less as compared to punense, where the genes are present in much larger number. So they are very similar at 6' rRNA gene sequence level, but that does not mean that physiologically they are identical. Physiologically, they could be very, very different. So that's the point which I wanted to emphasize here. So now looking at the differences at the global level, I'll quickly show this. this was a comparative study between Finnish population and Indian population. What we found is that this was the study done on teenagers, and what we found is, as expected, the microbiome is very different in Indian and Finnish population, and these differences could be dietary differences or because of the genetic differences. We cannot conclude that definitively, and there are certain genera which are very different. So as you will see again, the Megasphaera and Prevotella are the one which are predominant in Indian population, and in Finnish population you will find Bifidobacteria to be predominant. So the reason for difference in the microbiome composition is abundance of Prevotella and Megasphaera in Indian population, as against as Bifidobacteria and Glaucia are more abundant in Finnish population. We also did studies, I mean secretor status I'll skip, but mode of birth also we did study, and the point to be emphasized here is though in both the cases the mode of birth was shown to affect the gut microbial population, the way it affects is different in different populations. And another noteworthy point is that the mode of birth, that is cesarean birth or vaginal birth, this mode of birth, the effect of mode of birth continues even at the 13 to 14 year age. The effect continues, which in other words means that the way mode of birth affects the gut microbial population is lifelong. It is not that irrespective of the mode of birth, the gut microbiome becomes identical after a certain age. No, it's not like that. That difference continues throughout the life. That is a point which I wanted to emphasize. So now I'll just briefly talk about the disease, and here I'm first going to talk about diabetes. And we have already published one paper where we have seen that there is a dysbiosis in diabetic individuals, and then what happens is that in the healthy individuals, Prevotella is more abundant, but in the individuals, the Prevotella abundance goes down. And this is again very important because in other populations, Asian and Western population, it is shown that Prevotella is neutral. That is, whether you are obese or diabetic, the Prevotella abundance does not change. As against that, in Indian population, we have seen that in obese or diabetic individuals, Prevotella abundance decreases. So Prevotella is an indicator of dysbiosis. I mean, the decrease in abundance of Prevotella is indicator of dysbiosis, and higher abundance of Prevotella is indicative of healthy gut population in Indian population, in Indian individuals. So subsequently we did a more detailed study, and we studied a lot of biochemical parameters in the blood, lipid, vitamins, inflammatory markers, amino acids, and oxidative stress markers. And what we found was that the individuals could be classified into 4 categories: those healthy, non-diabetic, then there is a pre-diabetic category, and then there is third category is newly diagnosed diabetics. So newly diagnosed diabetics are the ones where there is impaired sugar metabolism has been detected, but no treatment is started. And this is a category which is very difficult to get. And then finally you have known diabetics which are under the treatment for more than a year. And you can make 4 distinct categories in the individuals which we studied. And what you will see is that there is a pattern. There is a pattern of gut microbiome which is different in all the 4 categories. And overall it indicates that in the healthy individuals there is a pattern which get disturbed when there is a diabetes. So you can see here, healthy individual, the green one, the microbiome is altered when there is a diabetes, and pre-diabetic you can see a trend. So healthy to pre-diabetic to diabetic, the microbiome had changed. I mean, you can see that here also. And for example, Prevotella, healthy to pre-diabetic to diabetic, Prevotella abundance has gone down. And when the treatment started, the Prevotella abundance again improved, but it has not come back to the level where it was earlier. So the diabetes status affect the microbiome changes from healthy to pre-diabetic to diabetic, and then after the treatment, the microbiome tries to come back to normal, but it is not as with the normal. But the important point to remember here is that this is a cross-sectional study and this is not a longitudinal study. So individuals which you are talking about here are different individuals. It's not the same individual that has been followed. So that is a point to be remembered. So this part, and what we also observed was that when we looked at the different biochemical markers, what we observed was that the Prevotella is an indicative of good marker. So because fasting glucose HbA1c, there was a negative correlation and there was a positive correlation with histidine and total antioxidant levels. So Prevotella, Akkermansia, they show a good positive correlation with healthy status, whereas there are some other bacteria like Megasphaera or Statella, they show positive correlation with diabetic markers and negative correlation with good markers like antioxidant levels. So there was a correlation between gut microbiome and diabetic status. And now based on this, I'll just come back to our proposed study. So based on this, we have now proposed to do a pan-India mapping of microbiome in different 4 biogeographic regions, 17 communities, and total 3,400 individuals we are going to sample. And I'll not go into the details. These are our participating institutes: NCCS, All India Institute of Medical Sciences, SRM University Chennai, IPSD Imphal, TDU Bangalore, and University of Pune. These are going to be participants. And we are also going to do genome sequence, collect information on dietary habits and the microbiome. And we hope to develop India-specific solutions from this study. So this is my entire group that has participated in this study. Of course, there are students working on different aspects other than microbiome. For example, Siddharth is working on, or Siddharth was working on AMR in effluent treatment plants. Snehal is working on polysaccharides from halophilic bacteria. Kusum was working on microbial communities associated with endolithic rocks. So they worked on different aspects. Satish was working on donor-like microbiome, but all other students were working on different aspects of microbiome. And these are my collaborators. Ashish Bavdekar, I collaborated with Indo-Finnish studies on infants. Govind Makharia, I collaborated on celiac disease, which I have not presented here. And Indo-Finland comparative study was done in collaboration with Dr. Seppo Salminen. Yadnik, I collaborated for diabetes studies. Dr. Ranade helped us in isolation of anaerobic organisms. And such a study is incomplete without anthropological background. So Dr. Shantanu Ozarkar and Dr. Zubekar were our anthropologists who advised us in the study. Thank you very much. And if there are any questions, I'll be more than happy to answer them. Hello. I can't hear anyone. Probably your mics are mute.
Are you able to hear me, sir?
Yeah, now I can hear you. Yeah.
I thank you so much for your presentation, sir. You have given a comprehensive overview of the microbiome in the global scenario and also in the Indian scenario, and the enormous amount of work that you have done especially with respect to the healthy Indian subjects and with respect to diabetes. Thank you for the wonderful presentation, sir. I think we have few questions.
Right, yeah.
So Dr. Harish Kumar has a question. Can it be possible to maintain the beneficial microbes in the gut by eating specific food?
Yeah. So that's what I mean. You can maintain healthy microbiome by eating healthy food, and what is meant by healthy here is uh, two things. One is your traditional food, in the sense that the food which is commonly eaten in the geographic area where you are located, that is always good for you, and the food in which is rich in fiber and less of sugar and fat, and in order to have increase the number of good bacteria in your gut, you should always eat traditional fermented foods, like whether it is dosa or idli or curd, whatever, you should eat that. And here again, this was another study which was done in our lab. I would also like to say that there was a study done in our lab on idli, and it was found that normally microbiologists would believe that once the idli is steamed, the bacteria in the idli would be killed. But we observed that some of the bacteria, and these are good bacteria, they survive whatever amount of time idli is steamed, they survive that. So which means that even steamed idli is a good source of probiotic bacteria, and idli has a very rich source of healthy bacteria. Again, I would say that a colleague of mine had done this study, and he had studied the flavor component of the idli and the diversity, and we found that diversity is correlated with flavor. So more the diverse microbiome in the idli is, more tasty and more spongy the idli is. So that's the way to maintain a good bacteria in your gut. Eat a lot of traditional fermented foods of the region, and diet that is rich in fiber and less in sugar and fat.
Thank you, sir. We have a question from Dr. Mahesh Patil. He says that now...
Yeah, I can read this question. It's about the gut microbiome and health link, right?
Yes.
Yeah. So there are many aspects to this question, but yes, it is now it's an established fact that your behavior is dependent on gut microbiome, and some of the things like anxiety, depression are related to gut microbiome and the way gut microbes metabolize your food. So that is now more or less established fact. And again, the issue which he has raised about the microbiome. So we have not yet gone into that, but we have published a study, and also another group from IGI has published a study where they have tried to correlate the Ayurvedic Prakriti with the gut microbiome, and there are some indications that in some of the Prakriti individuals, there is increased abundance of some specific groups of microbes. Both the studies have been published, but at least in our study, the drawback was the sample size was low, and in their study also, it was kind of biased towards one particular Prakriti. So there is a need to do a systematic study of well-defined Prakriti and the microbiome, and our proposed microbiome study has that component. We are going to have individuals with well-defined Prakriti and study their microbiome.
I think Dr. Ravisha has a question for you.
Yeah, his questions I cannot see here now. Somebody has to read it out.
Yeah, I'll just speak out, sir.
Yeah.
It was very informative. outside.
So,
Can we think of some smear or inoculation?
yeah, so those experiments have been done. Those experiments have been done. People have for the cesarean born babies, people have smeared the babies with the placental microbiome, and they have found that yes, it works. They do get healthy microbiome that way. So it works.
Has that been commercially practiced, sir? Now nowadays,
See, in India, these studies have not been done, but these studies have been done. All these studies have done abroad. Microbiome is a very, I should say, a very preliminary science in India. We do not have any data on even healthy microbiome across the country. So even FMT for treatment of some diseases, that has started hardly in last 2 years. So initially it started in all India Institute of Medical Sciences. Then at many places now people do FMT for IBD and colitis and such things. But these other explorations, we are far behind in this. There are no systematic studies to understand all these things.
Thank you.
Would you like to answer about what is the frequency of mutations in the microbiome and its induction with cancer?
See, frequency of mutations in the microbiome, I don't think that has been studied as to how the gut, how fastly the gut microbiome mutates and which mutation are retained. There was one study published some time back where it was shown that by the time you grow, I mean, from your infancy to your adulthood or and beyond, the E. coli in the gut has undergone so many changes that at the end it may be an altogether different organism. So I remember one such study, but otherwise this has not been studied. And coming to cancer, yes, today there are many studies which correlate cancer and the gut microbiome, and the largest studies have been done, of course, in colorectal cancer, and microbial community composition of individuals with colorectal cancer has been shown to be different from the healthy individuals. Affects or predisposes to colorectal cancer, again, that is something which is not known. There are a lot of studies in pancreatic cancers also. So microbiome and cancer is again another field where a lot of information is coming in.
Okay, sir. We have few questions with regard to the effect of the microbiome in times of the COVID-19 pandemic, and can probiotics help us increase the immunity?
Gut microbiome and COVID, there have been no studies. You see, COVID has come in last few months. Even today, we don't understand how COVID is affecting different microbiomes, whether nasal microbiome or gut microbiome. How COVID is affecting, we don't know. So anything talking about probiotic and COVID, it could be a very far-fetched this thing. But what I can say is from the published literature on other influenza infections is that there have been some reports where a nasal organism was shown to have protective effect on development of other type of influenza, including swine flu. So Streptococcus epidermidis in the nasal microbiome was shown to have shown to modulate mucosal immunity and in turn offer protection from other influenza. I cannot talk about COVID as such because such studies have not been initiated. But there are some proposals. I mean, I had a liberty to look at some international proposals, and yes, there are some proposals where people are trying to look at such things, looking at the nasal microbiome of affected, unaffected individuals, and then possibly develop a nasal probiotic. Because gut probiotic won't work here.
Thank you, sir. I think we have a lot more questions, but we are running out of time for the next.
Yeah, they are free to send me email. My email ID is there on my slide, so they are welcome to send me emails.
Yes, yes. I think I will send across the questions to you and then we can.
Sure, sure. Yeah, thank you.
Thank you so much for your
Thank you.
presentation.
Thank you. Thank you.
Now it is my pleasure to introduce the next speaker of today's webinar, Mr. Kumar Shankaran. Mr. Kumar Shankaran is the CEO of Lucin Limited and the designated partner in Gene Express LLP. He is a business leader with a professional experience of more than 15 years in healthcare research and delivery. In his current role, he is spearheading the growth and innovation in the microbiome domain for the Asian market. He is passionate about driving the paradigm shift from sick care to healthcare. Lucin Rich Bio is India's first and only microbiome-focused genomics-based company. I'm grateful to you, sir, that you accepted our invitation to deliver the talk on the topic, Role of Microbiome in Healthcare, the Different Vantage Points. Now I would invite Mr. Kumar Shankaran to present his talk.
Thank you, Dr. Catherine. Thank you for inviting me to give this talk. That was a wonderful talk by Professor Yogesh giving us an overview of the microbiome. They're doing some really great work in his lab. Today, my talk is going to be focused on the healthcare aspect of the gut microbiome. A lot in the last few years. I don't want to get too much into the basics of the microbiome, just one or two slides of it, and then I want to jump into the healthcare aspect of it. And later in the talk, where I want to spend a little more time is on the different viewpoints that people are having about the gut microbiome, and how we should view microbiome in general and gut microbiota in specific, and how that is actually impacting healthcare. So as mentioned in the previous talk, most of the microbes that are there in our body are located in the gut. Each individual has a unique gut microbiome, more like a fingerprint. But I'd also like to mention this, that the gut microbiome is transient. I mean, it also changes over a few months based on the kind of food and lifestyle an individual has. So unlike a fingerprint, which is constant, the gut microbiome, although unique to one individual, also changes with time. When we're focusing on healthcare, the aspect that most researchers around the world, most doctors around the world right now are focusing is on this dysbiotic state and trying to get the imbalance right. Now, do we know what the right balance of the microbiome is? No, not yet. I mean, I think there still needs to be a lot more studies that have to be done to understand what a normal microbiome or a normal microbiota would look like. But we do know a lot of the healthcare professionals are having right now is to correct or rebalance this dysbiotic state. I'm going to talk a little bit about inflammation in the next few slides, especially in gut dysbiosis and chronic inflammation. So when we see inflammation and damage in the epithelial layer, then we see a lot of both metabolites as well as bacteria going from the gut to other regions and from other regions into the gut. And this is now being seen as a major source for chronic diseases. Inflammation is a good thing. It is especially short-term and acute inflammation is good for a lot of healing. But as we get into chronic inflammation, as we abuse our body a lot more, we get into progressive destruction, and that inflammation is what could be causing a lot of diseases in our body. Um, in terms of the disease association, the microbiome is probably associated, especially the gut microbiome is now associated to a lot of diseases. The last count I did was about 72 diseases that the gut microbiome has been associated with. But there are very few diseases in which the causation pattern has been established. So we still do not know for majority of the diseases how the microbiota is actually contributing to the disease. We still do not know whether it's the change in the microbiome is because of a disease, or whether changes in the microbiome is causing a disease. Some of the areas where a lot of work that has been done, and where we are now beginning to understand the causation pattern, are mostly in the metabolic syndrome diseases, and in a lot of the cancers. I'll just run y'all through few examples of these diseases. Let's start off with atherosclerosis. I think this is probably the most well-established mechanism that has been found. I haven't gone into details of exactly which bacteria is producing what kind of metabolite, and how the changes in the microbiome affect it. But in the overview is, based on the kind of food that we're eating, based on the composition of the gut, flora, there are different metabolites that are being formed, which get secreted into our body, and that then causes problems in other parts of the body. And this is how this is one of the mechanisms that has been shown for atherosclerosis. Similarly, studies have been done to look at the microbiome between obese and lean individuals, and see the different effects that the gut microbiota has on different organs in our body. I think the most profound finding that has come through in all of this is the satiety signal. How the bacteria in the gut is actually signaling to the brain on satiety, and this signal is disrupted as people become overweight. There's a lot of work that has been done on diabetes, especially early stage type 2 diabetes. And like it was mentioned in the previous talk, I think it is now slowly becoming possible to detect diabetes at a much earlier stage. And we're getting to a place where we can even reverse early stage diabetes. We're not yet there, but I think there's a lot of science that is being done, which can prove to be through the gut microbiome, that we can get about the change in reversing diabetes. I think the gut-brain axis is probably the most talked about axis right now. Starting from anxiety, depression, going to Parkinson's, autism. Now there are a few studies that have even come out for Alzheimer's. So in all of these studies, what has been found is that there is a strong connection between the gut and the brain, and this is bidirectional. The brain influences the gut microbiota, and the gut microbiota influences functioning of the brain. In the cases that I have seen personally, I mean, we get a lot of individuals who come to us with symptoms of anxiety and depression, and the changes in the gut microbiota indicate that they could be playing a major factor in an individual being depressed. And by dietary changes and slowly over a period of time, there is no miracle probiotic or prebiotic that is available that you can just take it for 3 days and it will be cured. It is slow change in the microbiome through natural food sources, that can bring about the change, that can alleviate any kind of anxiety and depression. So there is a strong role that microbiome plays in depression. With respect to Parkinson's, also we've seen cases in which symptoms of Parkinson's have come down. I'm not saying that the disease has been reversed, but the symptomatically the individuals showing certain symptoms of Parkinson's have come down. I also wanted to touch a little bit about reproductive health and the microbiome. There's been a lot of talk about this. There's a lot of studies that are happening. Um, this is one tricky area where the role of the microbiome is not fully understood. Um, even placental microbiome, there's a huge debate between whether there is a whether there's a placental microbiome that actually exists or not. There are papers proving it. There are papers disproving it. So there is definitely a role that the gut microbiota plays, but it is still evolving science, and there is still a lot of studies that needs to be done, and especially a lot of studies that need to be done in the Indian context to understand the role of the gut microbiota in reproductive health, both from a expectant mother's point of view and from the expectant child point of view. Um, we keep talking about the microbiota and microbiome, and most of the time we talk about bacteria. Bacteria comprises major portion of the microbiota in the gut, but it is not only bacteria. It is there are a lot of fungus that are present. We've seen enough cases where there's dysbiosis in the fungal colony, which is actually causing problems in the gut-brain axis. There are more and more studies that are coming out where the microbiome, which is the fungal component of the gut microbiota, playing a strong role in a lot of diseases. And especially in the Indian population, we're also seeing different species of fungus being colonized in the gut. Not only bacteria and fungus, please let's not forget the viruses, especially the bacteriophages that are present in the gut microbiome. There was I'm showing one study that we were a part of. This is a Parkinson's study, and here you can see there is a clear differentiation between bacteriophages present in normal population versus bacteriophages present in Parkinson's disease. So virus also plays a major role. So it is not one type of organisms that play a role in the gut microbiota. I think it is the entire microbiota as a constituent that plays a role in health and wellness. So what exactly is the microbiome? I mean, this is a debate that has been going on for the last few years. There was an excellent paper that was published in June of 2019, where this question was posed. How we view the microbiota, right? How we view the microbiome also impacts on how we can help in healthcare. And there have been many views that have come out over the years. I have consolidated it into 4 brackets. And these are these views that I want to talk about extensively today to try and explain the pros and cons of each of these views. I do not have an answer to it. My opinion probably changes with new and new science that is coming out. I'm hoping with this talk that a lot of y'all will start asking these questions, and we can start defining what the microbiome is, especially from a healthcare context, much better. Let's take the organ view. I mean, let's think about the gut microbiome as an organ, and like we do for most other organs, what we tend to do then is uh, get a marker for how good the organ is doing, or how bad the organ is doing. Our way of doing this is a gut microbiome index. So we've come up with a score which ranges from -5 to +5, and depending on the composition of the microbiome an individual has, based on a lot of research that banged against a lot of research that has happened in the microbiome space. We give a score to the gut microbiome of that individual. So viewing it as an organ, you tend to give it a certain score, and based on that score, you're trying to correct any imbalance that are there. Now, the good thing about this is it is probably very, very simple to understand, very, very simple to practice. This is probably something that worldwide a lot of healthcare professionals are practicing, where they look at the gut microbiome, they look at whether it is dysbiotic or not, and based on that, give very specific inputs. So these inputs could be probiotics, could be prebiotics, could be nutritional therapy, could be any of this. But the aim over here is to correct some kind of uh, supposed malfunction that is happening with the organ. The challenge to this view is, unlike an organ, any other organ in our body, our microbiome is constantly changing. The number of species, the type of species, the number of cells, all of that is constantly changing. So since it's so transient, can we then call it an organ, or should we call it something else? So that is the question that is coming from the organ view. Um, this is probably a subject that has been talked about a lot in the last few months. Is, can we call the gut microbiome or any kind of microbiome in our body as an immune system? Is it part of our immune system? There is a strong axis between the gut and the lung, but there is significant work that needs to be done to prove any kind of causation or protective mechanism that the gut microbiota could have. But there is also studies that have shown that the microbiome, especially the skin microbiome, the gut microbiome, plays a big role in protecting our bodies from external pathogens. So it can be viewed as an immune system. Um, I mean, this is very good to theorize, but the question that all of us have, and to which we still don't have the answer for, is can the microbiome differentiate between self and non-self? Is this is it part of our body? Is it saying that this is an external pathogen which will harm the human body as a whole, or is it specific set of bacteria or fungus protecting its own niche in a particular area in our body? So that is the question that is coming up with the immune system point of view. Then there's this entire range of the holobiont theory that humans are more holobiont, and we have hologenome, and the hologenome concept coming into play, that we are a superorganism, and it is the genetic makeup of all our own genome and the microbial genome put together what constitutes a human being. Um, it's a very, very cool theory, resonates well with a lot of the younger generation. presents ourselves as some kind of a super being. Um, but but the question a lot of us have is this system, is this microbial system heritable or not? There is some kind of um, microbial passing on, where the mother gives the initial microbiome to an offspring through the birth canal. That theory is there, but um, is this a system that can be like how we pass on genetics? Is this system also passed on uh, from from parent to an offspring? Um, the the question in healthcare in this is, um, are we looking at the gut microbiome only as um, are we looking at the gut microbiome only as an organ, and are we correcting it, or are we taking it in in conjuncture with other metabolites, other genetic factors, the host genetic factor, the environmental condition, all of that into consideration, to act upon health. That is the question that is coming up with this. And the final view on this is the ecosystem view. Okay, the ecosystem view is very, very large. I'm just taking one very, very small example in this, where I wanted to talk about the kind of food that we eat influences our microbiome. Here, this is from a publication which looked at an apple which was produced through conventional farming, which is using pesticides, and apples which were produced using organic farming. And we see that the organic farming in almost all regions of apple have a much better diversity of the microbiome. The thumb rule as of today in the microbiome domain is, more the diversity, the better it is for you. So the it also our health, our gut microbiome also depends on the kind of food that we're eating. There's been an interesting experiment that has been proposed, which wants to look at the gut microbiome of food being prepared by individuals having different emotions. What happens, when you prepare food when someone's angry, as opposed to when someone is calm and having positive thoughts. Does that influence the kind of microbiome that we have? So the ecosystem view is a little grandiose. It takes into consideration a lot of factors uh, from our body, and all the things in our ecological system which is affecting our bodies. The although this is comprehensive, it is very, very difficult right now to measure any of these, and a lack of a clear boundary to see what the ecosystem is also impairs our objective when it comes to health and wellness. Yes, it's good to be in an ecosystem which improves the human microbiome drastically. But what is that ecosystem? How do we develop it? There is still a lot of work that needs to be done in that. Um, so to summarize, the implications of how we are looking at the microbiome affect the way you're looking at healthcare. Do you only look at it as a single component, give it the right kind of nutrition, and make sure that any kind of dysbiotic state is being cured? If that is happening, then you're probably ignoring the lifestyle habits, all the root cause for the change of the microbiome in the first place. If you're wanting to do that, is it the job of the healthcare professionals to look at all of this to try and give a very, very comprehensive solution? So these are the questions that are normally coming up in terms of how we look at the microbiome and how it impacts healthcare. But what is predominantly done both in India and outside India right now is looking at the microbiome as probably like an organ, and giving nutritional lifestyle and probably prebiotic probiotic recommendations to correct any of the imbalances that are there in the gut microbiome. Um, I briefly want to talk about the kind of work that we're doing at Lawson Rich Bio. So we started working on the gut microbiome in probably 2015. So we went around India collecting samples from different parts of India, looking at different kinds of nutrition, different kinds of geography, trying to map out the different microbiota based on the geography and nutrition. The Indian food is so diverse. We had to break down what every region was consuming, what is their natural food resources, and based on a lot of that, we came up with a gut microbiome test called as Buck Speaks. We've also done one clinical trial for this test. It now detects susceptibility. This is not a diagnostic or a prognostic test. It only detects susceptibility to about 15 chronic diseases. And what we do after this detection is provide nutritional recommendation. So our concentration is more on providing natural food and food sources to correct any dysbiosis in the gut. There is no miracle probiotic that is available that can be consumed that will change a diseased person's condition. In recognition of the kind of work that we've done, we've won the prestigious Frost and Sullivan work. All of this was done by scientists working in our team, and our mantra has been that if you can take care of your gut, it will eventually start taking care of you. This is my team. We're comprised of microbiologists, molecular biologists, computational biologists, and even statisticians. Thank you. If there's any questions, I'll be happy to take them. That's my Twitter handle, so you can tweet me. I'll also share my email address, and you can email me if you have any questions.
Thank you so much, sir, for the beautiful presentation and for giving us a perspective on the microbiome and its implications in healthcare, especially with respect to immunity, the holobiome concept, and also the possibility on the effects of the ecosystem on our microbiota. Thank you, sir, for the presentation. I think we have few questions. Okay, we have a question. How can we develop indigenous probiotics for the Indian population?
That's very difficult. I mean, we still have to map out a lot of the Indian population to figure out which of the bacteria is missing in the Indian gut. Also, please remember the gut microbiome is transient. So a probiotic might be relevant only at a certain point of time in an individual, and probiotics colonizing the gut is still a question mark. I mean, taking natural probiotics from fermented food. I mean, taking it through, like Professor Shaushay mentioned, through idlis or through curd or through our very own kanji. Through any of this will have a much better colonization potential than taking off-the-shelf probiotics. So there are two things that needs to be done. One is there is still a lot of research that needs to be done on the Indian gut microbiome to determine what kind of bacteria that is consistently missing to come up with a generic probiotic. This is a harder task. And secondly, I think it is a probiotic is only useful for an individual at a certain point of time in his healthcare journey.
We have another question. Is there any relation between gut microbiota and migraine?
There have been associations that have been done. The causation pattern is not yet established, but migraine has there is a link between the gut microbiota and migraine.
Okay, could you please correlate the gut microbiota and Parkinson's disease in your own words?
It's so there is. We still don't understand whether the gut microbiota is causing Parkinson's or not. That is something that we still don't know. But what we have seen with a few case studies that we have done is by trying to correct. So most of the Parkinson's patients have a very different kind of gut microbiome, which is going towards the dysbiotic stage or in chronic dysbiosis. By correcting those dysbiosis, by bringing it back into more eubiosis state, what we see is that the symptoms come down in Parkinson's. But this does not mean that the Parkinson's has been cured.
So we have another question regarding the non-vegetarian diet. So do you think the non-vegetarian diet could lead to distribution in the gut microbiome in a bad way or in the other way around?
It's very difficult to pinpoint a certain diet which could influence which could influence the microbiota in a bad way. But yes, having a lot of processed food, chemicals that are coming with the kind of food that you're eating. So it could be a fruit full of pesticides. It could be a meat product which is being processed. All of that influences your gut microbiota. It is also very important to eat local and to be seasonal. Especially with your fruits and vegetables. Overconsumption of anything, including meat, could be detrimental to the gut microbiome. So I wouldn't say that non-veg alone is a bad thing for the gut microbiota, but overconsumption of anything could be bad for the gut microbiome.
Is there any correlation of evolution in the change of the microbiota?
There's been interesting studies that have been done where the evolution has been checked, but it's too early to say anything about the evolution of the gut microbiome and of any kind of microbiome and evolution. There was a very interesting thought experiment that was done in 2019. I think that Reddit channel has been closed right now, but that was about time travel and especially the skin microbiome and how our skin microbiome is so different now as compared to what it would have been 200 years ago. So even if time travel is possible, our microbiome will not allow us to survive if we go back to 100 years. So that was a very interesting thought experiment that was done on, I think it was on Reddit about a year ago.
I think we can take one last question. Can the probiotics modify the genetically associated disorders?
Good. I mean, let's remove probiotics from it and let's answer it as, can gut microbiome change genetically associated disease? The partial answer I would give is there is an influence that the gut microbiome plays. This I'm saying with respect to the autism spectrum disease. One of the major portions of ASD is genetic in nature, aberrations in the human genome. But the gut microbiome can influence some of these symptoms that autism children display. Yes, I mean, there is a partial role that the gut microbiome is playing in genetic diseases as well.
Thank you so much, sir, for your patient presentation and also for answering the questions of the audience. Thank you.
Thank you. Thank you. Thank you so much.
I think we now come for the time for the last presentation for the day by Dr. Mukesh Kapoor. It is a pleasure for me to introduce the speaker. Dr. Mukesh Kapoor is the principal scientist in the Department of Protein Chemistry and Technology, CSIR CFTRI Mysuru. He completed his PhD from University of Delhi South Campus and has been serving as a scientist in CSIR CFTRI since 2009. He is also the recipient of CSIR Raman Research Fellow at the Michael Smith Laboratories, University of British Columbia, Vancouver, British Columbia. His area of research is focused on prebiotic action of mano-oligosaccharides and xylo-oligosaccharides in probiotic bacteria using genomic and proteomic approaches, deciphering of metabolic pathways involved in utilization of oligosaccharides in human probiotics using transcriptomic approaches, and deciphering the role of prebiotic oligosaccharides in modulation of gut microflora in diseased conditions, among other areas of carbohydrate enzymology and nanotechnology. I now invite Dr. Mukesh Kapoor to present his talk on role of prebiotics in modulating the gut microbiome. Over to you.
Yeah, good morning, everybody. Am I audible?
Yes.
Oh, yeah, thank you. So at the onset, I thank JSS Academy of Higher Education and Research for inviting me to give a talk in this webinar. And as Professor Yogesh Soche has said that there are some advantages and disadvantages of webinar, I will tell you there are some advantages and disadvantages of being the last speaker in a presentation. So the advantages are that I am aware, I mean the last speaker is aware that what kind of talk has been given by the other speakers, but the disadvantage is that I need to compromise on some of my slides because I don't want to waste the time of the participants in telling the same things. So what essentially I will do is to put less focus on something what has already been told and try to spend more time on something else. So let's begin it. I mean, so this has already been told that humans are having a very diverse and dynamic community known as a microbiota, and which is essentially shaped by the selection and competition, and essentially it helps in giving a protection from pathogenic microorganisms. And what essentially it does is to confer a lot of metabolic activities which essentially are not there in the human genome. And if you see this point that we humans are encoding only 17 enzymes for the digestion of food glycans. And overall, though we are having around 97 glycoside hydrolases, but out of them, almost 80, we are not having any direct function related to digestion. And we are just relying on around 8 enzymes and possibly some 9 enzymes for digestion. But on the contrary, if you see that what kind of enzyme composition and number of enzymes which are say coming from the microbiome, so you can see that it is enormous. So around 2169 enzymes are responsible for the degradation of plant cell wall glycans, and similarly you can see that a large number of enzymes are responsible for the digestion of animal glycans and some other substrates. And essentially, I also want to stress on one point that there is a large deal of interaction among the members of the gut microbiota, like the bacteroids, the lactobacillus, the bifidobacterium, and there is a kind of a sharing of the goods, because when we ingest the dietary fiber in form of say arabinoxyalans or chitosan, or there are many other things which I will tell maybe in my subsequent slides. But I want to stress upon the point that there is a lot of sharing of the resources, which can be public goods, means a bacteria is producing some enzymes which are degrading a fiber or a prebiotic, and it is being the resources of that, I mean the monosaccharides or the disaccharides are shared by the other bacterial members, but in other cases there is that is called as private goods where the bacteria is not sharing. So I will just rapidly go through these slides because most of these aspects have been told by the previous speakers, but I would like to stress upon the fact that a lot of this understanding about the molecular and cellular mechanisms for the gut hemostasis have recently happened because of the post-genomic high throughput technologies. And now what we are seeing is that alteration of gut homeostasis has been considered as one of the very important factors in several diseases which have been not seen till now, like IBD or celiac disease, obesity, and diabetes. And the previous speaker has already told about the brain-gut axis disturbances. So if you see that in the gut microbiome, it is a large intestine which plays a very important role because it inhabits a very high number of bacterial density, almost to the order of 10 raised to power 13. And this composition of the gut microbiome has already been told by other speakers also. It is very transient in nature. And there are many factors which are affecting it, including the mode of delivery, the feeding pattern. In fact, I would like to say that the breastfeeding is very important versus the formula feeding, because humans produce human milk oligosaccharides, and which are having around 200 different structures, and they are very, very important in the establishment of the Bifidobacterium infantis, say populations in the gut of the infant. And the same thing is actually missing in formula feeding, where actually the formulas are only having the galacto-oligosaccharides and the fructo-oligosaccharides, which are actually not specific for the Bifidobacterium infantis. Apart from that, there are many other factors which are responsible for the changes in the gut microbiota, which include the host genetics, the kind of diet in terms of, so westernized diet has been shown recently to have to result in a reduction in the diversity. So there has been, in fact, one more study that has shown that the Japanese people are having, say, enzymes in the gut, microbes which can degrade the seaweeds. So what has been shown that these enzymes, these genes have been taken up from the seaweeds what they are eating, and so the diet is actually playing a very, very important role. And of course the antibiotic use, we Indians are very used to go for the broad spectrum antibiotics, even in case, so we are not accustomed to go for a particular antibiotic. Normally we go for broad spectrum antibiotics, and that also affects the gut microbiota. So basically, it is the prebiotics which stimulate and maintain the gut microbiota, and the definition is there that prebiotics are basically non-digestible to humans, and they affect beneficially the host, that is the humans. And some of the so you can say that prebiotics can be a prebiotic oligosaccharides like galacto-oligosaccharide or a fructo-oligosaccharide, or some emerging oligosaccharides like beta-mannose-oligosaccharide, which my lab is working, or xylo-oligosaccharide, or derived from pectin. Similarly, dietary fiber can be of which are a little bigger in length, and they are like arabinoxylan, resistant starch, cognac mannan, pectin, chitosan, and these are some of the representative structures of oligosaccharides. So, as has already been told by other speakers, the potential health benefits can be summed up in three ways. One is immunomodulation, other is a metabolic effect, and the therapeutic effects of prebiotics when they nourish the probiotic population. So I will not go into the details further of that because other speakers have already given, but just like to stress upon the fact that these, like for diabetes and obesity, many papers have come and which has been shown at the molecular level that how the prebiotics are positively influencing are helping in the
Okay,
yeah.
Your slides are not visible.
Slides are not visible.
No.
Oh, sorry. 1 minute.
Not sure.
One minute.
Yes, sir.
So I have to press the share screen, right?
Yes, yes.
So, okay. One minute. Actually, I did it, but still it is not coming.
Can you click
One.
on the share screen option again?
Which one?
Share screen option at the
Yeah,
bottom.
yeah. I am pressing it. Share screen.
Yeah, select your presentation, sir, from the pop-up.
Yeah, that's how it is not coming. It is on my desktop only.
Okay.
So when I am 1 minute, give me just 1 minute. There is some issue. Shall I rejoin for the meeting? I think that may help.
Yeah, you can try logging in again, sir. I think we have some technical issue. I request the audience to please be patient for a few minutes. Another one shop, sir. Dhananjay. Can you hear me, sir?
Yeah, how? Yeah, actually, this Catherine, this one, like when I'm sharing the screen, I'm not
Okay.
using the desktop. How to go about it?
Can you come again?
Like when I'm sharing share screen, I'm pressing.
Okay.
This, it is not showing my desktop. It is showing like other things, iPhone, whiteboard, something like that. It is coming.
Okay. Is it not showing your presentation, sir?
No. Actually, yeah. It is not showing like from where I have to upload.
Your presentation is open, right? On the desktop.
Presentation, I can open it. 1 minute.
Yeah, you have to open it, sir.
Okay. I have opened it now.
Yeah, now you try the share screen again.
Yeah, it is same. I mean, it is not showing my presentation. I mean, it is just some other things are coming. Yeah,
Okay.
yeah, it is there now. Yeah. Can you
Yeah.
see it?
Yes, we got it.
Okay, so shall I restart?
I think you can come from the introduction slide, sir.
Yeah. This is visible now, right?
Yes, it's visible, sir.
Okay,
Please.
thank you very much for telling. Actually, I would have finished everything otherwise.
No issues.
Okay, thank you. So, so sorry, participants. It seems my slides were not visible, and maybe I can just brush up what I was trying to say. Is that the gut is inhabited by trillions of microorganisms, which we called as the microbiota, and this microbiota is shaped by selection and competition, and is basically responsible for the protection against the pathogenic microorganisms and enormous metabolic activities which the human genome is not having. But surprisingly, if you see that for the digestion of the food glycans, humans are only having 17 enzymes. And as you can see over here, that though we are having 97 glycoside hydrolysis in our genome, only 8 are responsible for the digestion of food and 9 are not having clear function. But rest 80, we do not have very known functions. But on the contrary, if you see the mini microbiome is contributing around 2169 glycoside hydrolysis for the digestion of plant cell wall glycans, and similarly very high number of hydrolysis for the digestion of animal glycans, other substrates, starch and glycogen. But in order to do so, there are a kind of interactions also which happen in between the various gut microbiota members. Some of these interactions are called as interactions for the public goods, means that a fiber what we have eaten is digested and the monosaccharides or the end products of that are shared by the various bacteria. But in other cases, it is private goods and a bacteria is not sharing the resources with other bacterial components. So I won't go into the detail of this slide, but I will just try to say that a lot of understanding has increased over the past several years because of advances in the molecular in the understanding of the molecular and cellular mechanisms for the gut homeostasis, which are driven through post-genomic and high throughput technologies. And now we are understanding that there is an alteration of gut homeostasis which happen in between several diseases like inflammatory bowel disease, celiac disease, obesity, diabetes, and brain gut axis disturbance, which even the previous speaker has told. So if you see in this slide, what I wish to say is that the large intestine is a place where a very high density of these of gut microbial symbionts exists with the highest density of 10 raised to power 13 bacterial cells. And so among the various factors which are affecting the composition, it starts from the mode of delivery, as one of the previous speaker has already told. In fact, the feeding patterns. So if you see that breastfed infants are having a very having a different or I should say the normal set of gut microbiota when we compare it with formula feeding. And one of the important factors which is responsible for this is the presence of human milk oligosaccharides in the breast milk, which is lacking in the formula fed formula milk, because they are complemented by fructooligosaccharides or galactooligosaccharides, which are not specific for Bifidobacterium infantis, which is the very first colonizers of the infant gut. In fact, westernized diet is also lacking fiber, and many studies have shown its influence on the gut microbiome, that it leads to less diversity of the gut microbiome. In fact, one of the studies for Japanese microflora has shown that consumption of seaweeds has enriched their microflora with genes for agarases and likewise enzymes which can attack the seaweeds. So similarly, inappropriate antibiotic usage has also been shown to affect gut to taking the broad spectrum antibiotics and not rather the specific antibiotics. So prebiotics are those molecules which are going to selectively stimulate this gut microbiota, and they can be a prebiotic oligosaccharide like a JOS, or a FOS like galactooligosacchariide or a fructooligosacchariide, or emerging oligosaccharides like beta-man oligosaccharides, in which my lab is working, and or they could be a dietary fiber like ribinoxylan, resistant starch, pectin, and chitosan. So these are some of the representative structures of the prebiotic oligosaccharides. And so if you see the health benefits of prebiotics and probiotics, and again not going into too much of details, the health benefits can be categorized into 3 types. It could be immunomodulation, it could be metabolic effects, or the therapeutic effects. So for example, in diabetes and obesity, the short chain fatty acids, which are very important molecule, and I'm going to discuss in detail in my subsequent slides. So they are the one which help in preventing the development and progression of the disease. And there is other aspects include the level of decrease in the level of the cholesterol or the low density lipoprotein, or stimulating the signaling of the satiation peptides like GLP-1 and the glucose-independent insulin-tropic peptide GIP. So, so we need to understand what is the interplay between the prebiotic-enriched diet and gut microbiome is actually dependent on the production or the fermentation of these fibers in the gut and leading to the production of short-chain fatty acids which act like a signaling molecule. So these acids are actually weak acids, and one of the things they does is to reduce the colonic pH, which actually prohibit the growth of the pathogenic bacteria. So majorly these SCFAs are of three types. It can be a propionate, acetate, or butyrate. And like butyrate is a very important source for the colonic epithelial cell and very important for the metabolic activity. So acetate is involved in enhancing the ATP production. Propionate contributes to gluconeogenesis. And if you see, there is a study for the European children, the bacterial counts, fecal bacterial counts of European children with a rural African village, which are almost isolated from the rest of the world because it's a kind of a tribe. And what has been shown, and they are actually eating more of fiber-rich diet. So the study has indicated that there was a significant difference in the gut microbiota of these two, say, selected groups. So coming to the further at molecular level, how these SCFAs, which are obtained after the fermentation of these probiotics and dietary fiber, how do they affect? So it is nicely shown in this picture, which I have taken from a recently published paper. So they affect the differentiation, they affect the promoting of the T regulatory cells. And these T regulatory cells are very important with respect to autoimmune diseases. When they reach to the portal vein, these SCFAs, they are important in the fatty acid oxidation and also for the activation of AMP kinases. And they also affect the increase anti-inflammatory cytokines, interleukins. And also there is a lot of say reports about how they decrease the incidence of colorectal cancer and by reducing the oxidative stress, and in case of IBD, that is irritable bowel syndrome, and by how they does it by activation of pro-carcinogen metabolizing enzymes like GST. So essentially how these studies are performed, because many students are attending that, so I will put this slide. So essentially, if we need to understand that how this microflora, which is comprising the gut microbiota, so we need to do some co-culture studies in order to understand the role of a particular, say, candidate probiotic, prebiotic, like it could be a fiber, it could be a oligosaccharide. So essentially, many people, and actually this is a very recent concept nowadays which is coming in order to get the real-time picture that how, because normally such studies are done in isolation when we are feeding a bacteria a particular oligosaccharide and trying to see the effect. But nowadays, these are the upcoming studies in many journals where they are trying to show that how a bacteroid or a bifidobacterium and a lactobacillus, if they are grown in a co-culture, how the utilization will happen. And so these bacteria, like bacteroids, many of their gene clusters have been studied in detail, and they are called as PULS. PULS stands for polysaccharide utilization loci. So this is one which is used for the degradation of xylem in case of bacteroids. Now, these bacteria, which are comprising the gut microbiome, how do they sense the presence of these, say, oligosaccharides in the lumen? So they use a variety of sensing machines, I should say, molecular machines. Like, for example, the extra cytoplasmic function, ECF sigma, anti-sigma. So the binding of the oligosaccharide over here releases the anti-sigma factor, and which actually releases the cognate ECF sigma, which goes and interacts with the RNA polymerase. There are some other receptors called as CESR-like. Then the other type of receptor is called as a hybrid two-component system, which actually is comprised of two domains. That is, one is the histidine kinase domain, and other is the response regulator domain. So the binding of an oligosaccharide over here will result in the autophosphorylation of a conserved histidine residue over here in the HK domain, and which will lead to a phosphotransfer to the regulatory response domain over here, and then leading to the further interaction with the DNA. So and leading to further set of say a cascade of reactions. So essentially these bacteria if you see at the molecular level, they are actually producing a battery of enzymes which are highly specific in their interaction and for the capture of the oligosaccharides or the polysaccharides which are there in the gut by producing some enzymes which are extracellular in nature and some of them are periplasmic in nature. So if you see this beautiful picture which I am showing is for Bacteroides ovatus, and you can see that there are certain enzymes that is called as endoxyloglucanase, which are endo-acting enzymes. So they will cut this large polymers into small pieces. So two of them are produced. One is a GH5A, one is a GH9A. So after that, there will be, as I told, that there is the carbohydrate sensing machines are there, and so this will help in the further interaction and also the transport through the membrane. And so when they come inside the periplasmic space, some other set of enzymes like arabinofuraanosidase. So in this diagram, you can see that the star and the blue dot, blue circle, they are representing the various monosaccharides. So arabinofuraanosidase will act on the arabinofuraanosidic linkage. After that, a glucosidase will act, that is the GH3A, GH3B, and the alpha xylosidase will act to remove the xylose residues. And finally, it will come to a beta galactosidase, which will chew it down to the monomer. So you should see that it is not that simple that we are taking a fiber. It has occurred over a period of thousands of years, which has helped in shaping the humans right now what we are, because of the specificity of the enzymes which are being produced by these bacteria. So now recently some studies have shown like how the co-culture is happening, as I was discussing previously, that how the co-culture of Bifidobacterium adolescentis can happen with Bacteroides ovatus, and you can see that both of them, if they are in a monoculture, are not able to grow on a xylem, which is as complex as birchwood xylem or a wheat bran xylem or a corn xylem. But when it is a co-culture, there we see a lot of growth over here. So actually my lab works on mannans, and so mannans you can see in nature, it's a very important hemicellulose, other of course being xylem. So mannan it can be found in fenugreek, it is found in guar gum and ivory nut, so the locust bean gum, and of course in the plant cell wall. So mannans are of various types. It can be a beta mannan, it can be a galactomannan when it is decorated with galactose residue. It can be a glucomannan when we are having a glucose. It can be a galactoglucomannan also. And there are some yeast mannans which are called as alpha mannan. So there are two types, beta and alpha. And so there are variety of enzymes which degrade mannans, like endobeta mannanase is the main enzyme which acts on the beta mannosidic linkages. And yeah. So actually, so we wish to understand that how these beta mannans are utilized by, say, the intestinal, by the gut microbiome member. And this work was reported for Rosoboria intestinalis, which is a very important gut microbiota member. And as I have told previously, that a battery of enzymes and transporters are responsible for the uptake of and degradation of the carbohydrate fractions which are undigested by humans. So as you can see over here that there is a transporter which is there in orange, and then there is a CBM binding domains which will help in the binding of these polysaccharide fractions. And then once it is internalized, there are various type of enzymes which you can see over here that which will remove these decorations, which can be it can be acetyl decorations, so over here, and or it can be some glucosidase like GH3A, GH3B over here. And finally, we are going to get the monosaccharides in form of the say, mannose and glucose, which are actually fed to the glycolytic pathway and again lead to the production of a short chain fatty acid, say over here like butyrate. So when these mannans are also fed, say in our diet, they also undergo a similar pathway. So as I have told about Rosoboria intestinalis, so our interest in my lab is to work on lactobacillus. So the pathways in lactobacillus for the utilization of prebiotic oligosaccharides like fructooligosaccharides and galactooligosaccharides are pretty well known. But in case of mannooligosaccharides, the pathways are not known. In fact, we have shown almost 3 years back that that mano-oligosaccharides can be utilized by lactobacillus. But what was not known that how these mano-oligosaccharides are being transported inside the bacteria, what are the transporters, and what are the enzymes which can degrade these mano-oligosaccharides. So we started our work on the purification of these oligosaccharides by various techniques like mass spectrometry and NMR. And what we found, maybe I'm just skipping these slides. So what we found that 3 types of mano-oligosaccharides like mannose, galactose mannose, and galactose mannose are produced from locust bean gum by the mannanase, and which was in fact having an unusual pattern that it was producing a galactomano-oligosaccharide, as you can see over here, which is very rarely produced. And so we have shown that these mano-oligosaccharides in purified form are improving the growth of lactobacillus, various species of lactobacillus, and they produce short chain fatty acids. But and we have also characterized them, also these mano-oligosaccharides, by using various techniques like FTIR and also by thermogravimetric analysis, because it is important to know at what temperature these oligosaccharides are stable. So we found that they are stable at very high temperatures, around 300 degrees Celsius. Interestingly, we also found that they are decreasing the survival of the pathogens like E. coli, Salmonella typhi, and Listeria monocytogenes. So we have also produced these oligosaccharides on a continuous basis using magnetic nanoparticles, and so this is just a characterization, so I'm just skipping this point. So you can see over here some beautiful pictures of these magnetic nanoparticles with the immobilized enzymes, which can be used for the production of the mano-oligosaccharides. So you can see, so these type of mano-oligosaccharide, DP2 means that 2 units of manos, 3 means 3 units, and 4, and subsequently. So till this point, we have established that mano-oligosaccharides can be produced and can be purified, and they are able to show prebiotic activity for lactobacillus. Similarly, we have shown mano-oligosaccharides from guar gum, which is also a very important source from Indian context, because India is the biggest producer of guar gum. And over here also we have shown the prebiotic potential of the guar gum oligosaccharides in improving the growth of lactobacillus species and inhibition of some pathogenic bacteria. So, but the main question still was hovering over our head, that how it is possible, because the enzymes or the transporters are not there. So we did a more systematic study, and we saw that in case of Bacteroides ovatus or in case of Bacteroid theta atomicron, there are enzymes which are shown in the genome, and so, and of course they are responsible for the cleavage and for the cleavage. So, but when we saw the lactobacillus genome, we were not able to find any genetic loci which can be which is responsible for the utilization or the hydrolysis of beta mano-oligosaccharides or beta mannans as such. So only thing what we found, so in fact there was no endo-beta mannanase or a manosidase, so it was very perplexing for us. So, but we saw that there was a GH36 alpha galactosidase, and which can cleave the galactose links, which is found in galactomannans like guar gum or locust bean gum. So we cloned this enzyme and expressed this protein also, and surprisingly, we have not found any activity of this enzyme on a galactomano-oligosaccharide. So it was not able to cleave it. So it was very surprising for us, and so we did some other studies on that. Maybe I will come back to that later on. So we again went back to our biochemical experiments. We did simple HPLC experiments to again prove our point that yes, Lactobacillus plantarum is able to utilize these mano-oligosaccharides or not. And so this is a very nice HPLC chromatogram on which you can see that at various hours, what is the amount of mano-oligosaccharides which are available. So what we found that they were utilized quite quickly, even before the sugars by Lactobacillus plantarum, as you can see in the in the D picture, sorry. So we went now one step further, and we did the transcriptional analysis of beta mannose utilization in Lactobacillus plantarum. So the strain that we have chosen for this study was WCFS1, which is a known human probiotic lactobacillus. This we got from Netherlands. And so what this study has, these are the volcano plots, and the data was deposited in GOE Omnibus. So what this data suggested us that by the heat maps and the volcano plots, that there are two loci which are getting upregulated in the presence of monooligosaccharide. One is the sallobiose utilization loci comprising of 6-phosphobeta-glucosidase and the transporter, that is PTS23C. And there was one more, say, loci, that is the oligosucrose utilization loci, which is getting, say, upregulated. So we corroborated this data with real-time PCR assays, and we really found that both the transcriptional analysis and the real-time PCR data was matching, and we were seeing a lot of upregulation in specifically in these two loci. So the data can be seen over here. So so we did one more thing, that was a comparative operon analysis with other members of the Firmicutes. So, but it was very surprising for us to see, as can be seen in this picture, that the gene content of these bacteria was quite varied when we see with respect to sallobiose loci or the oligosucrose loci. Even when we saw the position of 6-PBG, which was fairly consistent in all these lactobacilli or other Firmicutes, but the orientation was quite different when compared to WCFS1. Similarly, the raffinose 1 hydrolase, that is the AGL2, we were only found in Lactobacillus plantarum and Paracasei and not in other bacteria. So it was very intriguing for us that how these bacteria, which are actually belonging to Firmicutes, are having different orientation and genes with respect to these two loci. And finally, we have proposed a mechanism that how these manobios, so we have proposed that this manobios is actually taken up by the PTS23C and then acted upon by 6-PBG, that is 6-phosphobeta-glucosidase, while the monoligosaccharides, which are having substitutions of galactose, are taken up by PTS1 and subsequently acted upon by raffinose 6-phosphohydrolase. So in order to prove the transcription analysis in real-time PCR studies, we went one more step further in order to delete the loci, and for that we have done actually three techniques. One is a homologous recombination, followed by insertion deletion of the genes, and then cloning of the sallobiose operon. So the homologous recombination was done using a Kralox-based system. Insertion deletion was done using PET28 and PSAT-A, and the sallobiose operon was cloned in PET-duet vector. So, however, unfortunately, the homologous recombination did not resulted in any positive clones. So this approach failed. In case of insertion deletion, also we were not successful because we have not got any clones with respect to that. However, the expression of sallobiose operon expression, we were partially successful because we were able to get the 6-PBG expressed, but however not in case of PTS23C. And so this gene was cloned and expressed, and this enzyme was not showing any activity on the synthetic substrate what we were having in the lab. So what we actually required was a substituted beta-mannoligosaccharide. So which was phosphorylated. So essentially, though this data is negative, but we got to know that the phosphorylation is a must for this enzyme, and we are taking up this now further. So coming to the other part of the GH36 that I have shown earlier, we were able to produce a prebiotic DP3, that is aminotriose from this enzyme. So this is just purification and characterization of that, and we have established through mass spectrometry and through NMR studies that it is a aminotriose which this enzyme can produce. And this aminotriose was again prebiotic for Lactobacillus plantarum and Bifidobacterium adolescentis. So finally, as I come from a food research institute, so our idea was to show that whether these beta-mannoligosaccharide which we have produced in the lab and are essentially prebiotic in nature, can they be used in the preparation of symbiotic ice creams? So we made such ice creams, and this is just a general preparation method. And interestingly, what these ice creams, what we have found, as you can see in this graph, that the survival of so what happens is, like when you put a probiotic in ice cream, so what happens is you are going to store it at very low temperatures in minus 20 degrees Celsius. So what happens at the time of when a consumer is taking it, so the amount of bacteria will be pretty less because the temperature of storage is quite harsh. So what we have shown in this study was that not only the bacteria is being held by beta-man oligosaccharides in during its stay in the ice cream during the storage period, but it is also helping the bacteria to survive in the gastrointestinal stress, because the stomach acid or the pH which is there in the intestine is not congenial, say for the survival. So the ice creams were really able to say support the bacteria over here. So in all, I would just like to now conclude by telling so my lab has basically focused on the gut microbiome, specifically targeting the lactobacillus. And now, so this work was done by Deepesh, and who is now going to be a postdoc at University of British Columbia in a short while. Right now, he's in the Indian Institute of Science, and she is Priyanka. She is actually following the work on Bifidobacterium. And so other students over here, they are working on different aspects. And so I am really grateful to the support which has been given to me from Department of Biotechnology in New Delhi, from CSIR, from SERB, and of course from institute funding to really help us to reach this far. And so I thank you all for especially the JSS for giving me this opportunity to share what kind of knowledge we have gained till now in the area of prebiotics. And I will be really glad to take up now few questions.
Thank you so much, sir, for the wonderful presentation and for giving us an overview on the role of probiotics in the gut microbiota. I think we have a few questions. I think I would like to ask a question first.
Yeah, sure.
So you specified on the role of mannans in Lactobacillus plantarum in your laboratory.
Yeah.
I would like to know, have you seen the effects of these mannans on the other microbiota in relation with Lactobacillus?
Yeah, so we have till now we have focused on Lactobacillus, and we have tested various strains of Lactobacillus. And now with my other PhD student that I was telling, Priyanka, she is focusing on Bifidobacterium and Bacteroids. And so essentially what happens is, like, Bacteroids are not going to take up these oligosaccharides in a way that they are the generalized degraders. So they will take these oligosaccharides in competition with Lactobacillus or Bifidobacterium, and but they are more going to go for the fiber part. I mean, where we are having the DP more than 10. So, but in case of Bifidobacterium and Lactobacillus, and what we have seen also, that they are having specialized transporters for the oligosaccharides, which can be taken up from the lumen. But yeah, we are we have not gone beyond that, but yeah, really we wish to do. So in fact, I have got a recent project now from ICMR in which we are going to have an in vivo picture of that. So maybe when we meet next time, I can give you more data with respect to mannan utilization by the gut microbiota.
Sure, sir. So with respect to a common man's view, how would you suggest, what is the kind of diet we should consume which would introduce these prebiotics to enhance the number of, or the diversity of the gut microbiota?
I think India is really blessed with a lot of traditional foods, especially like which are made using millets. And because actually we are really restricting ourselves to by eating many polished foods, right? Even rice we are eating without bran, and even the atta what we are eating nowadays, it is you can say like it is just like maida only. So the idea is to eat more fiber, because it is the non-digestible fiber which is the key, which holds the key for the successful say maintenance of the gut microbiota. So I think one should eat a lot of fiber-rich diets, and we should definitely include fruits and vegetables also, because they also provide us fiber. And one should try to avoid foods which are not having fibers, just for example like bakery items. And so the idea is to go for a more balanced diet, It's not like that you have to only eat fiber, you have to take other nutrients also. But yeah, one should try to avoid highly processed foods. That's what I feel.
Thank you, sir. We have two other questions. I don't know if
Yeah.
it is relevant, but I think it can be open for the other panelists too.
Yeah, sure.
To avoid the value addition side effects of bifidobacterium in probiotic product, what are the parameters that need to be considered?
Sorry, come back again with respect to?
Okay, to avoid the value addition side effects on bifidobacterium in probiotic product, what are the parameters that need to be considered or to check?
So what essentially I think they are trying to ask is like you are making a functional food with respect to bifidobacterium. Am I correct?
Yeah, I suppose.
Yeah. So I think it is very important that when you are making a functional food, one need to make sure that the count of bacteria which is going to reach to the intestine should be very high. It should be more than 10 raised to power 6. The kind of processing now being which is being going to be carried out for making a functional food should not be deleterious to the bacteria. And bifidobacterium, for that matter, is anaerobic in nature. So one has to be very careful if you are going ahead with bifidobacterium in a functional food.
What is the homology with Saccharomyces cerevisiae of fructokinase?
Actually, we have not worked on fructokinase, but yeah, Saccharomyces cerevisiae is a very important, say, yeast, and is a definitely important part of the microbiome. But we have not done any studies with respect to the homology modeling or homology studies with fructokinase from Saccharomyces.
Thank you so much, sir. I think one last question which
Sure.
I would like to put across to all the panelists is, I've recently read a lot of articles on the correlation between diet, exercise, and the microbiome. So what is your perspective or your take on that?
I think all of them are very intricately related, because diet is the most important factor, because that is a kind of a nutrient which is going to feed the say trillions of microbes which are residing in this microbiome. So diet is very important, and exercise definitely helps us in keeping us fit. So I guess the sedentary lifestyle, but yeah, there are no independent studies which have been done to show, okay, a person or is doing exercise and the person is not doing exercise, and how it is really affecting the gut microbiome in general. But yes, as poor exercise is definitely going to affect the health, and it is not simply exercise, it is a sedentary lifestyle, which is the most important factor over here, that a person is having a sedentary lifestyle and is uh, and that sedentary lifestyle is going to affect say various metabolic parameters, and we have seen in several studies that how deleterious the sedentary lifestyle is in contributing to the metabolic syndrome. So I guess all these factors are intricately related. You cannot ignore exercise, you cannot ignore your diet, and of course if you are taking care of both of them, the health will be in a fine shape.
Thank you, sir. I think I like a view from Dr. Kumar. Do you think we can separate both exercise and diet when we are studying the diversity of the microbiome?
There were experiments that were done in 2016, which kind of showed that exercise independent of diet changes the gut microbiota. And I think there were further experiments which were trying to prove that where you're exercising also matters as to how the gut microbiota changes. What we have seen with individuals who take part in national level athletics, when we see their gut microbiome data, that's almost the other extreme. I mean, they're putting their body through so much of stress, and there's so much of inflammation that is happening because they have to train really hard. The number of actually beneficial bacteria count in their gut is actually coming down. So when we're seeing it with athletics, it's almost like a bell-shaped curve. Sedentary lifestyle is bad, and you will have a poor gut microbiome. As the exercise, as activity goes up and exercise goes up, I mean, there is a good microbiota. But if there is strenuous activity that is happening and very very hard training that is happening, then also you see a drop in the microbiota. So that's what we've observed.
Thank you, sir. I'd like final comments from Professor Yogesh sir regarding in the times of COVID-19 pandemic. What are your suggestions to the general audience to maintain healthy gut microbiota and general your health? And can we use our traditional medicine knowledge to maintain our gut microbiota? Okay, we have a last question. How to maintain the microbiota when we are under antibiotics? I don't know. Mukesh sir, would you like to take that question?
Yeah. So we know that antibiotics are going to affect even the probiotic microbes, and I think since childhood, some therapy that has been given is to always take lactovate, right? So, so idea is to take some spores of lactobacillus so that the amount of the probiotics. But yeah, I think there. I don't know whether this is the correct forum to tell, but somehow like we are always used to our being prescribed a very broad spectrum antibiotics, even if we are having a simple throat infection, and where it is not necessary. So I think it should always follow in a way that that we should see which type of microbe is infecting and then followed by the antibiotic. That is one measure that can be done. But apart from that, yeah, I think because of so much of now interest and research which is coming in the area of gut microbiome, I feel that there in the coming time we should see more personalized medicine which are enriched with some gut microbiome members apart from lactobacillus, because that's one of the members, residing members, not all. So that's what I feel with respect to antibiotics.
Thank you, sir. Okay, I think we can wrap up the webinar here. So I'm very happy, and it is my pleasure to propose the vote of thanks. I extend my gratitude to the Registrar, JSS AHER, for permitting us to conduct this webinar as part of the Special Interest Group and from the Department of Microbiology. My heartfelt thanks to the Dean, JSS AHER, Dr. Balasubramaniam sir, and the head, Dr. Ravisha, for his constant support and their encouragement for the conduction of this event. I express my heartfelt gratitude to the speakers, Dr. Yogesh sir, Mr. Kumar and Shankaran, and Dr. Mukesh Kapoor for kindly accepting my invitation and presenting a beautiful talk. I owe the success of this webinar to all three speakers. Thank you so much, sirs. Special thanks to the Chief Information Officer, Ravindra sir, and the IT department, JSS AHER, for helping us organize this webinar and for their constant support, without which today's event would not have been possible. Lastly, I wish to thank all my colleagues, students, and the audience for participating in this event and helping it become a grand success. Thank you all. Lastly, I would like to apologize for any technical glitches. Yes, sir.
No, I just said thank you.
I apologize for any technical glitches that you might have experienced during this webinar. It was a great session to have all these speakers with us. An important announcement for the participants, which they have been asking me. Please do fill the feedback form, which will be sent to you on your email, after which you will receive the e-certificate. Thank you so much for joining us. Have a good day and stay safe. Thank you so much.
Thank you.
Thank you very much.
Thank you.
Thank you. Thank you.